we discovered that there were pockets of cancer cells that are enveloped by very dense layers of this connective tissue, the stroma, that prevented blood vessels from getting in there. And so those cells inside these pockets would become hypoxic faster. Some tumor cells became so hypoxic, they just were not responding to the radiotherapy. But in some of the tumors, once you got that treatment dosing right, you could actually make the tumor response much better. Hi, I'm Dr. Ralph DaCosta. I'm a senior scientist at the Princess Margaret Cancer Center here at the University Health Network. I'm also an assistant professor in the Department of Medical Biophysics at the University of Toronto. I built a laser myself in my garage when I was a kid. I was tweaking with this and tweaking with that, and one day I see this red beam of light come out, and I screamed. I'm like, oh my God, it works. That's what got me really interested in optics and the mathematics of optics, and then theoretical physics, trying to understand quantum theory and what is a photon, which is a unit of light. I can see the power of light being harnessed for medical applications. So my first job was a summer student in Brian's lab, and it eventually became my PhD work, was to invent and optimize fluorescence-based endoscopic imaging systems. Fluorescence is a very unique property of materials where if you illuminate that material with a very narrow band of what we refer to as excitation light, that band will cause excitation of the molecules at a atomic level. And what happens is those molecules take this blue energy and the electrons sort of jump up to an excitation state. Then when they collapse, they give off energy, which is in the form of light, and that's fluorescent light. Each of these different biological components that makes up our cellular and our tissues have different excitation profiles. I can use a certain wavelength that only creates fluorescence of the collagen and then flip to another wavelength that only creates fluorescence in those other molecules. And by doing that, using this sort of tunability, you can optically separate the different colors of fluorescence light that come out off the tissue. So we can track the cell in red, and when that cell starts to feel hypoxia, it also emits a green color. So the cell starts to look red and green at the same time. So it ends up looking more orange yellow. So we started to learn how these different layers of biological components, the tumor cells, the vessels, the stromal compartment, the blood flow, the hypoxic component, etc., how all of this is tied together, and we can actually see it right inside the living system and watch this biology occur. And we've learned so many new things. What was really mind-blowing to me was that here's a kid who loves physics and math, builds a laser, builds a confocal microscope, and now is trying to solve a really, really big problem that affects people around the globe. I love the instrumentation, but I really loved working with the clinicians and the patients. And so that became a real mission for me, to try to invent things that we could improve patients' lives and understanding of biology, and cancer biology in particular.